chapter 16, carbohydrate. And so these are the different basic carbohydrate types. We have monosaccharide, glucose, fructose. Monosaccharide only have one sugar molecule. Disaccharide, two monosaccharide linked together. And also the oligosaccharides, 3 to 10 monosaccharide linked by the glycosidic bond, that's oligosaccharide. And polysaccharide, that would be the chains of the linked monosaccharide unit. Okay, so these are carbohydrate types. Then the carbohydrate. And you look at these two functional group. We have this aldehyde, and now be the aldose. And then this is ketone functional group. I'll give you the ketones. All right. So that the difference is here. You see this carbonyl bond here with the C double bond oxygen, carbon oxygen. Then in the end, we have the hydrogen, and here we have CH2OH. So this is the ketones, and this is the aldehyde. Okay. So aldose, that'll be the monosaccharide containing the aldehyde group. So it's COH. And then the ketones, monosaccharide containing a ketone group, right? That's the aldose and ketones. And then the number of carbon atom in the main skeleton, right? So if there's 3 carbon, that's a triose. And then 6 we have a hexose, right? And then here is COH, that's aldose, the hexose. And then this one, CH2OH, that's the ketones, right? And this is glucose, all right? And this is fructose. So monosaccharide are classified by their number of carbon atom, and the triose will be the simplest carbohydrate monosaccharide, and the glyceraldehyde here. Glyceraldehyde, right? Have a sterol center, is it as a pair of an enantiomer and enantiomers, right? That's a mirror image stereo isomer. So like these two here, all right? So on the top is the glyceraldehyde. In the bottom, that's the left glyceraldehyde. And later on, the next few slides, that would explain, all right, the right and left. So this is a visual projection. So this is glucose. So this is visual projection. And the glucose, right? And this is fructose. Here's the aldehyde. This is ketose. And then this is the D-glucose, D-fructose. And all right, so and then here is to explain how to determine the D and L, right? So that always you have to put the most oxidized carbon on the top. Most oxidized carbon on the top, and as you can see here, this carbon is the most oxidized carbon. So put it on the top, we have COH, right? And then the chiral center, the farthest from the most oxidized end, right? So this one is the furthest, so that'll be in here. And then this, if this is on the right hydroxy group, right, on the stereo center, this is stereo center, is on the right of the stereo center, then it's a D. If it's on the left of the stereo center, here, hydroxy group, then it's L. Okay, so that's how you define the D and L. So put the most oxidized carbon on the top, and then the furthest chiral center, all right, and then look at the location of hydroxy group. It's on the right, then will be a D. On the left is L. Okay, So that's stereo-centered, right? And enantiomers are also called the optical isomers. Okay. And this is visual projection of monosaccharide, right? So this is most oxidized carbon. This is chiral center on the right. That's a D, all right? This is most oxidized carbon on the top. And then the chiral center, the furthest, the carbon here is on the right. So it's a D glucose. And this one also is a D fructose, okay? And the most common sugar are in the D form, right? And in L form, look at this chiral center, hydroxide group is on the left, so it's L. So the visual projection always put the most oxidized carbon closest to the top. So the D monosaccharides, you have to write it in the visual projection, then the hydroxy group on the right of the chiral center, then it'll be D. If it's on the left, the chiral center will be L. Okay. And then in here, so for example, from here, the carbon, the most oxidized CHO on the top, and then chiral center on the right, hydroxy group, so it's D, right? And then also if you go down the list, right? So here, the chiral center, and then hydroxy group on the right, this one on the right, and this one on the right. Okay. And glucose is the most important sugar in the human body, right? Has many common names, can be a dextrose or blood sugar. And under physiological condition, glucose exists in a cyclic hemiacetal form. That means the carbon number 5 hydroxy react with the carbon 1 aldehyde group. Okay, C1C5, so it'll be here, hemiacetal. The hemiacetal, that means this alcohol OH here react with the aldehyde carbon here, all right? So you remove one hydrogen here, this oxygen connect to this carbon. That's called hemiacetal. So C1, C5, all right? C1, C5, so this oxygen, you can see here, C1, C5, right? So that's how they react, okay? C1, C5 hemiacetal. And then when they form this cyclic form, this is pyrene, okay, pyrene. And then for this Haworth projection, this hydroxy group can be different, can be in trans side with the CH2OH or the cis side. If it's cis, all right, this hydroxy group is this CH2OH, then that'll be the beta. If it's trans, all right, this one and this one is trans, that's alpha, all right? And the alpha D, all right? D is because this chiral center, number 5 carbon here, hydroxy group is on the right, so it's a D glucose, all right? D glucose. And then when they form this reaction, this hemiacetal, okay, the COH and this number 5 OH here, okay, then they form this hemiacetal, and then you will look at this hydroxy group, okay, the hydroxy group either in the trans position with the CH2OH or the cis, right? And that give you alpha or D. Okay. So the monosaccharide have the hydroxy group or the carbonic group in the same molecule and exist almost entirely as 5 and 6-member cyclic hemiacetate. And then anomeric carbon, the new stereo center resulting from the cyclic hemiacetate formation. So this is alpha D glucose, right? Alpha D glucose. Alpha D glucose here. Originally the hydroxy group is here, right? And this up, it's a D. And then this carbon we have with this number 1 and number 5, right? We will form the hemiacetal, and they give you the alpha. Okay. Because this is here and this is here. If this one here, the hydroxy group is on the other side here, and this is also this side, this is cis, so it's a beta. And that's the Haber's projection. Okay. So alpha mean it is trans, right? This is a hydroxy group from number 1, and this CH2OH, all right? This CH2OH in number 5 carbon here. These two in trans, that'll be alpha. In cis, that'll be beta. And then this is alpha D glucose, okay? There's D because hydroxy group here, and alpha is because this hydroxy group here and this CH2H on the other side. So it's in trans. If this hydroxy group is on the left side and this is also on the left side of cis, so it's beta. And then also it's D glucose. And the reducing sugar, so that means one that reducing oxidizing agent. That's called reducing sugar. So when the oxidation of a cyclic hemiacetal that can give you a lactone. So when we use the oxidizing agent, okay, as pollen, and then silver will precipitate as a silver mirror. Okay. If the anomeric carbon are involved in the glycosidic linkage, then there would be a negative pollen reagent test. And if another anomeric carbon is not bounded and is free, there will be a positive pollen reagent test. And this is just to show you that the reduction of this carbonic group, if you reduce this carbonic group here, all right, then it becomes artichoke. So you reduce this, so you add the hydrogen here. So that becomes sorbitol. And for here, xylulose, you reduce this carbonic group, then it becomes like this, then it's a xylitol. Okay, that's the reduction of the carbonic group of monosaccharide. And the phosphorus ester, so are particularly important in metabolism of sugar to provide the energy. So here, so basically you can see they form this phosphorus ester. They transfer the phosphate group from the ATP right to here, right? Transfer to here, so like it is PO3, the phosphate group. So that becomes the beta D glucose 6 phosphate, and then release the ADP. And the glycoside, a carbohydrate in which the hydroxy group of the anomeric carbon is replaced by the OR. Okay. Is replaced by the OR, it's a glycoside. And so glycosidic bond, that's a bond from the anomeric carbon to the OR group. Okay, that's from here. And then see here, meso. Okay, this is the meso group here. So this is a meso alcohol, and then they replace this hydroxy group. They remove the hydrogen and put the meso group here. And that's the glycosidic bond. Okay. So the glycosidic linkage you can take various forms. The anomeric carbon of one sugar to any of the hydroxy group, another sugar to form an alpha or beta glycosidic linkage, right? So like this one is alpha 1,4. This is number 1, all right? This one is number 1, and then 1, 2, 3, 4, and this 1,4. It's a 1,4 glycosidic bond. And this is the beta 1,1 glycosidic bond, all right? So this is 1,1, and this is 1,6 alpha 1,6 glycosidic bond. So these are 3 commonly seen glycosidic bond, and then that will give you the now we will discuss these sucrose, lactose, maltose. They give you this linkage, all right? This linkage. So for lactose, we have beta 1,4 glycosidic bond, all right? This is beta 1,4 glycosidic bond. Sucrose, we have the alpha 1,2 glycosidic bond, and maltose is alpha 1,4 glycosidic bond, right? So that's the glycosidic bond to form the disaccharide, okay? To form the disaccharide. And then the fructose is also called a fruit sugar. Can be found in honey, corn syrup, fruit. Is the sweetest of all sugar. It's ketohexose. So this is a D-fructose, D-series hydroxy group, all right? It's on the right of this Charles center hydroxy group, okay? And then the 1,5-hemiacetal, then you have this alpha because this is in trans position and this in cis position, so this alpha D fructose, beta D fructose. And then the galactose is the principal sugar found in the mammalian milk. Okay, that's called galactose. All right, this is D-galactose, and then alpha D-galactose, beta D-galactose. Okay. And then this is different orientation between the glucose and galactose. You can see the difference in location of these hydroxy group at the number 4 carbon. Okay. And the ribose and the deoxyribose. It's here, this is ribose, this is deoxyribose, right? Ribose, deoxyribose. And the Benedict reagent. So basically the aldehyde group, like here, the aldehyde group of the aldose can be oxidized by the Benedict reagent, which is the alkali, the copper solution, right? And oxidize this. Yes, Hadel, you cannot hear me. So basically, you can use this Benedict test to measure the glucose in the urine, right? Benedict test. So it's about the the, the glucose in your urine. So you can do that kind of test, and then the color will change from the blue color to the red-orange color, right? And the reducing the sugars, all the monosaccharide and the disaccharides, except the sucrose, are reducing sugars, except the sucrose. Okay, everything be the reducing sugar. So a sugar can undergo the oxidation reaction, right? Glucose oxidation, right? So that's why they were reducing sugar. They can be oxidized. And the glycosidic linkage are responsible for the bonding of monosaccharide to form oligosaccharides and the polysaccharides. And these are just some important oligosaccharides. So we just mentioned the sucrose is glucose, glucose and the fructose is alpha 1, 2, glucose and fructose. Lactose is galactose and the glucose. And maltose will be the glucose and the glucose, right? Glucose. And glucose, galactose, glucose and fructose, right? That's these three different important disaccharide. And then the polysaccharide, so when many monosaccharides are linked together, and one example is cellulose. That's the major structural component of plants, especially wood and the plant fiber. So the D-glucose to 2,800 D-glucose unit per molecule, joined by the beta-1,4 glycosidic bond. See, the 1 is 4, okay, beta-1,4 glycosidic bond. And then the amylose here. So it's like a 4,000 alpha D glucose unit joined together as a 1,4, right? Alpha 1,4 glycosidic bond. Okay. So this is the amylose. And then aminopectin, which is a highly branched. So you can see the branch. There's a branched polymer consisting 24 to 30 units of D-glucose joined by this 1,4 glycosidic bond. Okay. So that's the polysaccharide. And then the major structural component of the exoskeleton of the invertebrate are chitin, right, in the insect. That's the chitin. So you have acetyl beta D glucosamine, right, like this. They join the beta 1,4 glycosidic bond. And then bacteria cell wall. Bacteria cell wall, basically you have this peptidoglycan, and which is important now from the gram-positive, gram-negative test. For those cells that has a very has the thick glyco, very thick the cell wall structure. Okay. The right? Because this Na and Nag, they form this peptidoglycan. And this peptidoglycan basically that give you the difference in the cell wall property. Then you can use the Gran's test to determine it's a gram-positive bacteria or the gram-negative bacteria. If the cell wall has this Na and Nag and the very thick peptidoglycan cell wall, then will be gram-positive bacteria. Otherwise, it would be gram-negative bacteria. So that's these beta-1,4-glycosidic link these NAM and NAG, all right? So the glycosaminoglycan, which is the polysaccharide based on repeating a disaccharide, where one of the monomer is an amino sugar and the other has a negative charge due to sulfate or carboxylate group. Okay, this is heparin. Okay, heparin is natural anticoagulant. This heparin. And the hyaluronic acid, that's a component of the various human of the eye and the lubricating the fluid of the joint here. And this is the keratin sulfate, the component of connective tissue, right? So these are the glycosaminoglycan. So basically the summary, the polysaccharide formed by linking the monomer sugars to the glycosidic linkage, and the starch and glycogen are energy storage polymer or sugar. And later on, when Dr. Father discussed those breakdown of these polysaccharide, then you will learn how the animal cells storage the glycogen, and then how they use the glycogen. And cellulose and chitin are structural polymers, and polysaccharide, important component of cell wall in bacteria and plants, especially in bacteria like the peptidoglycan that can be used to determine the gram-positive or gram-negative bacteria. Okay, so that would be for the